Hello audience, welcome to my channel. Today we are discussed about the phylogenetic tree and how we analyze the phylogenetic analysis, applications and brief demonstration on how to use the MAGA X software and with important points in interpreting a cladogram. So we discussed about today side by side the brief introduction and the application and the demonstrations. So keep watching the whole video so you will get the maximum benefit of the demonstration. So the phylogenetic used for the infer-evolutionary relationship using the nucleotides or amino acid sequence that's coming after the sequencing. So samples are grouped based on the similarities and differences of DNA or protein sequence. So the application is used in the classifications, forensic science, epidemiology, as well as the conversation. So for that purpose, we are using different type of molecular markers, proteins as well as genes that are specifically linked with based on the chloroplast as well as the mitochondrial genes that have the specific identity, so we can use these genes and after that we can sequence it and make the phylogenetic tree and compare it with the already NCBI database that we have. So for that purpose we will use the MAGA 10 software that is called the molecular evolutionary genetic analysis. So it has a different versions. So currently we have the updated one, the 10 as well as the 11. So meanwhile I am talking about the MAGA X. So we can use different sequence coming from different genre and we will blast it, and after that we have a specific sequence. We can use this sequence and use in the MAGA. So for that purpose we will make a FASTA file of the sequence that we have a specific. We will export it from the desktop sequence, and after that we will align the exported sequence, inspect the alignment, after that the trim access sequence, and finally we will save it the final alignment file into the MAGA format and construct the phylogenetic tree using different protocols. We can have the maximum likelihood as well as the neighbor joining methods. So here I am discussed about a little brief introduction of the experiment. Suppose we have the bacterial strains and we have the groups and we have the description, and after that we have the accession numbers. Here we have some our own sequence and some of the sequence that is already submitted by different countries scientists or researchers to the NCBI. So we can blast these sequence and check the similarity of the sequence, and the maximum similarity that we have, it means that we have the similar sequence into the database. So here we go for the uh, alignment, edit the alignment. In the edit alignment you can see follow the protocol that I am using in this video, so it will be easy for you. For that purpose go to the create a new alignment. After the new alignment the pop-up will appear, is your DNA sequence or the protein sequence, so you can click on the respected uh, pop up, and after that, if you have the sequence in the FASTA file or the query with the GENBANK, so you can directly import it from the GENBANK source, as well as you can import your own sequence that is present on the desktop version. If you have the sequence on the NCBI, so directly give your accession number here and go to the search, so it will be automatically appear into the mega file. And here you can see this is one of the gene of 16S ribosomal RNA partial sequence for one of the sequence. It is present in the NCBI. In the next step, this is the input labels. So all of the accession numbers and the names and the other things will be appear, and you can select the OK button. And if you want something editable, so you can be changed here. So it will be appear the same tag or the number into the mega file as a reference. So you can see here, here is all the sequence be here. So when you have import the file of the sequence or the FASTA file, after that you can make the alignment by the cluster W or the muscles. It depends upon the sequence that you have. After the alignment, you can see these colors. So these colors, it represents the matching position of all the sequence into the of your sequence. Some of the sequence is not matching, so it gives the dots or the gaps. In the next slide, you can see here, it will be appear for you. So you can see here, these are all the sequence that is not similar, so it giving you the condition by this way. So from here you can select and delete or trim your sequence, and only the sequence that you have align with your all of the sequence, so you can put it there. So here's the align DNA. So these are the parametric metrics that we are using for the alignment. So make it as default and go further. Here you can see very clearly some of the sequence in the color format and some is not. So similarly you can find this sequence in the forward side and as well as to the tail side that is not similar. So from here you can delete or remove the sequence one by one, or select it and go to the scissor or the cross button, so you can easily remove this sequence from the forward and from the tail side. Upper image show the forward side and lower image show the lower side. So we can trim and remove the sequence very easily. Here you can see from the forward and from the tail side the dissimilar sequence that is not aligned is removed. So after that you can save your file from the file option and export it into the MAGA format. From the MAGA format you will give a specific file name and the destination folder so that you can find easily. After that you go for the phylogeny and select the maximum likelihood at the neighbor joining. It depends upon type of analysis that you need. So after clicking on the phylogeny, so you can see here, click on the options on the specific analysis. So it will give you a file upload position. So the file in the previous session you saved it into the MAGA format. Open the MAGA and upload this file, and after the uploading, so these parameters appear on your screen. So most of the time we are using the 1000 bootstrap. It's giving the percent value of the substitution of the nucleotide sequence that we are using, and after that the distances that we are using, and collect data from all the sites. So it represents these specific conditions. Make it okay, and after that you can see it takes some time because due to the length of the video I just shortened the things, and after that it's appeared like this. So you have the options. You can change your phylogenetic tree into the longitudinal type, circular type, and you can save your picture into the JPG or PNG, PDF, as well as the other things. Even you can change the colors, the font type, size, and other things. Many things we can edit after the confirmation of the thing. So in this session I will describe how to describe the cladogram. You can see here we have three similar genotypes are the strain that is present in the one group, and we have the second clade called Pseudomonas suzerae, and you can see here they are making one group and the other is the second group, and we are using one out group as a reference group. So we will put this reference group is making and separate in the whole phylogenetic tree. In the third clade we have the Chevenille algae that we are using as a third group. So you can see very clearly that we have three types of groups that we are using in our phylogenetic analysis. This is the thing that I'm talking about previously. That's the type of the phylogenetic tree you need, either circular, longitudinal, or the rectangular type. So from here you can change the thing very easily, make it color, make it more attractive, and change the things as you want to need. So here is the confidence interval and the bootstrap value. So we are most of the time strongly spotted that have the value more than 90%, are well spotted 70 to 90%, and weakly spotted to 50 to 70%, and not spotted less than 50%. But it depends upon the contribution, similarity or dissimilarity that is present among the clones or the stems that we have. So this is all the descriptions and the results how we can analyze our phylogenetic tree based on the clones, outgroups, and different type of clades that is making in the picture. So here we are talking about the scale bar that is appear downside. So scale bar indicates the number of substitutions per sites. So this is also we call it sequence divergence. In our case it is the 0.020. So 0.02 mean is equal to 2 base difference for every 100 bases. So when we align the sequence, it means that if we have the 100 bases sequence, so after 2 base difference for every 100 base sequence disappears in this data. So if we have the value is 0.05, 0.01 or zero point 0.06, so you can see that represents that. Then it is the 2 or 5 or the 10 base difference for every 100 bases of the divergence from the original one. So as a whole, I am talking about the summary. So molecular phylogeny is used to elucidate evolutionary relationship, especially among prokaryotic organisms including viruses. Phylogenetic analysis has become an invaluable tool in discovery of new species, R strains, and in epidemiological study. Furthermore, with phylogenetic studies we can also use in the genome-wide analysis for different type of proteins that is formed.